Welcome to Introduction to Ecology, Lesson 9. Today we're beginning a new unit on populations. Now back in the first lecture, we actually defined a population as interacting individuals of a single species living in the same place at the same time. Today's lecture, we're going to talk a little bit more about how we define populations and how we measure and characterize populations in order to be able to talk about them and talk about their properties and the important aspects of their ecology. So again, one of the key aspects of populations and why ecologists are interested in studying populations is because the individuals within the population interact with each other. And there's a number of ways that individuals in a population can interact. One obvious way is with interbreeding. So different members of a population will mate and produce offspring. The other big way that individuals in a population interact is through competition. And this can be competition for a number of things. Food, if you're a plant, it might be sunlight, other resources, mates. So that picture on the right is two male elephant seals that are having a battle in order to win primary mates or good mating grounds, the best mates. And then nesting sites or even just places to live, shelter. It depends on the population what is limiting and what they compete most strongly for. But in general, in most animal populations, there is strong competition with other members of the population. So, on its surface, the definition of a population seems pretty simple. But if you think about it a little more carefully, it can get a little confusing. And one of the challenges is understanding what is an individual. So if I think about a fir tree in a fir forest, it's not that hard. So there's my individual fir tree, and here's my fir forest, which forms my fir population, my population of fir trees. But what if those weren't fir trees? What if those were aspen? And in a previous lecture, we learned that aspen can actually grow by cloning themselves. And so a whole big forest stand of aspen may actually be one sort of genetic type, exactly the same genes in all the trees in that whole forest. And so then the question becomes, is a tree an individual? And is the forest a population? Or is the whole forest actually an individual? Because again, if that aspen has reproduced clonally, it's reproduced by copying itself, then every single tree is genetically identical. And so maybe that whole forest is actually an individual. So clonal reproduction is fairly common in the world. It occurs in plants, in corals, in sea anemones, hydroids, some insects. And it even happens in larger, more complex animals like frogs, fish, and lizards. Now, fortunately, ecologists have run into this a lot because it's so common, and so they've come up with definitions for defining these clonal populations or these clonal individuals or whatever they are. The first definition is genet. And a genet is the product of a single fertilization event. So it's all of the clones that are part of the same genet. So if I have a plant and I cut off all sorts of leaves and I plant them all and they all grow up into new little plants, none of those new little plants started as a result of fertilization. They all started because I cut a piece off the plant and started growing it. And so they're all genetically identical. And those are all part of one genet. A ramete is the actually or potentially independent members of a genet. So unlike you, if I cut off your arm, you'll probably bleed to death and die. If I cut down one tree in a genet, the whole forest doesn't die. In fact, I can cut down another tree in the genet, and the whole forest doesn't die. And I can keep doing that until I'm left with one single tree. And so that tree is the ramete. It's the independent individual organism that may be genetically identical to the other organisms around, but it sort of acts as a unit that can live alone. So if you get rid of all the others, it could still live alone. All right. So that's a genet and a ramete, and that helps us to sort of understand the components of clonal groups, clonal populations, clonal individuals. We still have that problem though. What do you call an individual and what do you call a population? And actually there's no good answer to that with clonal things. And what you call a population and what you call an individual often depends on the question you're asking and the time scale you're asking it at. So if you're thinking about evolutionary time scales, very long time scales, the genet might be the most appropriate level to focus on because you're interested in changes in the frequency of different genes or changes in genotypes within a population. And so it doesn't make sense to look at all the different ramets because they're identical. There is no difference in the genes. There is no difference in genetic makeup. So it's hard to ask evolutionary questions if you call all the different members of a ramet a population. On the other hand, if you're thinking about an ecological scale, the ramet may be the most appropriate level for defining a population because I might be thinking about how forest tent caterpillars interact with ramets and how they defoliate different trees. And in that sense, it doesn't matter that the trees are genetically identical. What matters is how each individual tree is interacting with the population of tent caterpillars. And so again, with clonal populations or with clonal individuals, exactly what's an individual and what's a population is not super well defined and really often depends on what question you're asking. All right, that's the very basic definition of a population. Now let's think about how we can characterize populations, or what are the different properties of populations. And the first two main properties when I'm thinking about populations are distribution and abundance. So the distribution is the geographic area where individuals of a species are present. So here's a distribution for the bald eagle. And you can see that there is some area where the bald eagle is only present in the summer where it breeds. That's the blue. There's some area where it's present all the time, all year round, and that's the orange. And there's some area where it's only present in the winter where it sort of goes during the cold weather further north. So that's sort of a description of the distribution of bald eagles. And if you look at that distribution, bald eagles should be found all the time in Toronto. And I don't know why, but when I was a kid, I loved bald eagles. I lived in Canada. It wasn't my national bird, but I loved bald eagles. I really, really, really wanted to see a bald eagle. And I had Audubon's Book of Birds. And in the Audubon Book of Birds, you could look in the back of the book and you could see these maps that showed the distributions of the different birds. And so I looked at the bald eagle map showing the bald eagle distribution, and I saw it should live in Toronto. And I couldn't understand why, if the bald eagle lived in Toronto, I never saw one. But it turns out distribution is only one part of the story about a population. Abundance is another big factor. And it turns out abundance is the number of individuals of a species in a given area. If you look at bald eagles in Toronto, in fact, if you look at bald eagles in all of Ontario, I was living in Toronto up until the mid-90s, and what you'll notice is that in all of Ontario, there were less than 100 bald eagles up until about 1995. And so it's not surprising that even though the Audubon Bird Book said I should be able to see eagles in my hometown, I wasn't able to see them because their abundance was so low. It was actually low because of a number of factors, including DDT that they had used in the 70s. All right. So obviously defining population abundance is really important. It's one really important measure or important metric for characterizing what the population is actually like. So there's a number of different ways we can study population abundance. One is population size, and that's the number of individuals in a population. So we might say, okay, what's the population size of sparrows in Clemson? And so there's my city limits of Clemson, and I got my sparrows there, and I can count them up, and I can say, well, there's 11 sparrows. There's 11 sparrows in Clemson. But then you might think, why did we draw our boundary at Clemson? That's kind of weird. What's so special about Perimeter Road that we stopped counting sparrows when we hit Perimeter Road? And in fact, if we looked at a larger area, the outskirts of Clemson, what we see is that there's actually more than 11 sparrows. And it's even worse because some of those sparrows that are sometimes in Clemson city limits can also drift over and move outside of city limits. And so sometimes population size is not the best measure of abundance. And instead, we frequently use density, population density. This is the number of individuals per unit area. And so rather than 11 sparrows, what we could say is, well, there are actually 2 sparrows per kilometer in Clemson. And what you'll notice is that even if we look a little bit outside the city limits, it's still about 2 sparrows per kilometer, no matter where we are. And so sometimes population density is a bit more robust to sort of variation in how you define the area where you're studying your population. All right. So there's sort of another distinction when you're thinking about abundance, and that's the distinction between absolute abundance and relative abundance. Absolute abundance or absolute population size is the actual abundance. Relative population size, on the other hand, is the number of individuals at one time or place relative to the number at another. And I'm going to give you an idea of, I think we all can get an idea of or know what absolute population size might mean. I'm going to show you what relative population size is. So Audubon has been conducting the Christmas bird count since 1934. And what happens is people go outside on Christmas Day and they count birds. And they count all the birds they can see. And so you can imagine some birders going out in 1934. And they might count cardinals. 1, 2, 3, 4, 5, 6. Now, even if there were many, many, many birders out on Christmas Day, they probably haven't counted every single cardinal. There's some cardinals they likely missed because there weren't birders in that place, or they weren't watching when that bird flew by. So they don't know the absolute population size. They just know that they counted 6 cardinals. And that's important because fast forward to the future, say 2018, and we can have people doing the Christmas bird count, and they can also count cardinals. And maybe they get 1, 2, 3. And again, that's probably not all the cardinals in the area. They've probably missed some. They haven't seen them. But what they can say is that the population in 2018 was half as big as it was in 1934. And that's a relative population size metric. So we don't know that it was 6 cardinals in 1934 and 3 in 2018. In fact, it could have been 8 cardinals in 1934, but they didn't happen to see 2 of them. And it could have been 4 cardinals in 2018, but they kind of missed one. But that doesn't matter because it's still correct that there are twice as many birds in 1934 as they were in 2018. Same thing. Maybe there were actually 12 cardinals in 1934 and they happened to miss counting half of them. But as long as they also happen to miss counting half of them in 2018, it's okay. The relative population size is still correct. And so relative population size is typically easier to measure. It's typically what ecologists can measure. And is very important because it can show you variation. It can show you that a population is going up or down. It can show you that a population is bigger in one area, maybe in sort of a forest, than it is in another area, maybe a city. So again, a lot of what ecologists work with is relative population size. Very rarely do they manage to count every individual and know exactly how much is there, although sometimes they try. Frequently, it is the relative population size, which is also very informative. All right, so we've defined distributions and we've defined abundance. But what determines distributions and abundance? Why are some organisms only found in one place and some organisms really abundant in one place and really rare in another? There's a number of different factors that determine distribution and abundance of an organism, and we've talked a little bit about this before. So a big one is habitat suitability. That's not the only one, though. There's also historical factors and dispersal. All right. Within habitat suitability, a number of different aspects can influence where certain plants, animals, fungi, other organisms are found. We've seen this figure before. This was where researchers tried to predict the locations of different plants in Australia based on abiotic factors, based on things like climate, soil, pH, salt concentration, nutrients. And we saw that they did pretty well. And so what that tells you is that the physical environment, the abiotic conditions, go a long way in determining who can live where and how abundant organisms are in the places where they live. But that's not the only thing that matters. Biotic interactions also determine habitat suitability. So even if you have the perfect temperature and the perfect precipitation and the perfect wind speeds and the perfect soil nutrients, everything like that, even if that's all perfect, some organisms will not grow in those nice conditions because they're lacking biotic interactions or because of existing biotic interactions that prevent them or exclude them from those locations. So important biotic interactions that can influence where organisms are found. One of the first is mutualistic partners. A good example is the yucca and the yucca moth. So the yucca relies on the yucca moth for pollination, and the yucca moth relies on the yucca for food and reproduction. And so you will never find yucca moths living someplace where the yuccas can't grow, even if there's no physical conditions that would kill the moths themselves in those locations. And you will never find yuccas growing where the yucca moths can't exist, even if there's no sort of differences in rainfall or temperature or wind that prevent the yucca from growing there. The yucca needs to have the yucca moth in order to live where it does. Another good example, again, this is sort of one of those areas that I'm interested in. I love ants. And so a good example of mutualistic partners determining where organisms are found is what's known as devil's gardens. And these are interesting areas in tropical rainforests where there's only one species of plant. It's sort of like a clearing with just this one species of plant, and then all around it, the rainforest kind of is all around it with all the other different plants. And for a long time, people didn't know why these areas were sort of devoid of all the other things that grow in the rainforest, why there was only one type of plant in these devil's gardens. But it turns out ants are actually controlling that. The ants are actually killing all the plants except for this one species. And they're saving the one species because that's actually the plant that they live on. And so, again, because of what these ants are doing, it's determining where this very specialized plant that the ants live on is found, and it's also excluding all these other plants that would otherwise grow in that place but can't because the ants keep killing them. Another biotic interaction that can influence where things are found is herbivores or grazing. This can be mammal herbivores like voles or deer. It can also be insect herbivores, a lot of different herbivores out there. And so this is an example, for instance, what you see in this slide, where frequently scientists study the effect of herbivores on where plants can grow and how much plants can grow by doing what's known as exclusions. And so in this example, the scientists excluded voles from one area and they allowed voles into another area. And what they found is that the plants that the voles ate grew to really, really, really big heights and high abundances in the areas where the voles were excluded, but they were really kind of short, stubby plants and low density in the areas where the voles were not excluded, where the voles were allowed to eat them. By contrast, they found that all the other plants the voles didn't eat grew to much higher heights and much more abundant in the areas where the voles were not excluded, but they did not get as dense and as big in the various areas where the voles were excluded. So again, herbivores can determine where certain plants are found, and that again is a biotic effect. Predators, of course, very similar to herbivores, can determine where their prey is found. And we actually try to hijack this when we're gardening. So you can actually buy ladybugs to eat your aphids. And the idea is that you are trying to make your garden unsuitable to aphids that are killing your plants because you are introducing predators into your garden. Sometimes it works, sometimes it doesn't. Another biotic interaction that can influence where organisms are found is competition, or the presence of competitors. So this is a great example, a classic example, where barnacles grow in different locations, different heights above the low tide. And what you'll notice is that there are blue barnacles growing down lower, closer to the water, and there are brown barnacles growing up top. And it turns out the brown barnacles could actually grow everywhere along that whole stretch. But they don't because they get outcompeted by the blue barnacles at the lower, closer to the tide line. The blue barnacles, however, are not able to get too far up. They are not able to get all the way up to the high tide, And so that leaves a little region where they don't exclude the brown barnacles, and the brown barnacles kind of live up there. So again, this is an example where the blue barnacles that compete with the brown barnacles actually determine where the brown barnacles can live. Finally, another biotic interaction that can matter to where things are found is parasites or pathogens, the presence of parasites or pathogens. I think a good local example is in the Smokies. So there's a lot of animals we have extirpated from the Smokies, which means we've kind of hunted or basically in other ways gotten rid of them. And scientists have been doing a lot of work trying to reintroduce some of these species into the Smokies. So they've actually managed to reintroduce elk, and you can actually go about 2 hours north of Cherokee and you can see the elk. Scientists also tried to do the same thing with wolves. Wolves used to live in the Smokies, and you probably all know that they don't anymore. But at one point, scientists and park managers tried to reintroduce the wolves, and it didn't work. And the reason it didn't work was because in the time between when the wolves were extirpated and when people tried to reintroduce them, canine distemper virus had arrived in the park, and it arrived partly via domestic dogs, partly via coyotes. But regardless, the presence of this pathogen in the park actually made it impossible for the wolves to be reintroduced, to successfully develop a population in that area. All right. Now, we've talked about abiotic factors and biotic factors as if they're two separate forces. Often, though, biotic and abiotic factors interact to determine a species' distribution and abundance. And so it's not necessarily just parasites or pathogens or just competitors. It's a whole mix of things that sort of work together to determine where things can live and how abundant they are in those locations. The final aspect of habitat suitability, something we don't often think about as much, is disturbance. Disturbance is an abiotic event, whether it's fire, drought, flood, wind, that kills or damages some individuals in a population. And this basically makes it easier for other things to grow there and harder for the things that are damaged. And so the frequency and intensity of disturbance can actually influence what grows where. And you can see this in this map across the US, where in the locations where we have low rates of disturbance, we actually have forest and woodland type habitats. And in the areas where we have high rates of disturbance, for example, this is specifically fire disturbance, we have grasses and shrubs. All right, so that's habitat suitability. That's a big determinant of where things are found, but it's not the only determinant. So we also have historical factors and dispersal interacting to determine where things are. Dispersal is the movement of individuals, and that can be the movement of seeds, animals, spores, even pollen. And dispersal links patchy populations together. So dispersal limitation happens when a species' limited ability to disperse, its limited ability to move, prevents it from reaching areas of suitable habitat. And we've talked about this a little bit in the past with fire ants. And we said, fire ants didn't used to live in South Carolina. Back in the 1700s, there were no fire ants in South Carolina. And it wasn't because the habitat wasn't suitable for fire ants. It was because fire ants had no way to disperse to South Carolina. They actually lived in South America, and there was no way for them to move all the way up to South Carolina. When humans, however, helped them with that move, then all of a sudden they found this great suitable habitat, and they've done really well ever since. So in general, species tend to come from regions where they originated, and sometimes they come from regions where they found refuge. So they may have evolved someplace else, but then they managed to disperse to a certain location, and then an event forced them away from those locations into what we call refuges. And frequently when we're talking about refuges, it's with respect to glacial periods. And so, it'll be sort of lower latitudes where the ice never got to, and that will be a refuge for certain animals. And then when the ice melts, the animals will come back and disperse away from that refuge and move further north, for instance, or further up a mountain. So again, what matters in terms of where species are found is the continent where they evolved, any land bridges that allowed them to move from one sort of continent to another, continental drift that brings different continents close together and then moves them far apart, and then of course ice ages. And then how far they get beyond those regions, whether they actually get from one continent to another, or whether they actually move up after an ice age, up away from the refuge and towards other places, depends on their ability to disperse. And so the picture on the right shows the dotted line where we had ice free. Anything below that, the glaciers never got to. And what you'll notice is that depending on how fast an organism can move per year, or a population can move per year, determines how far away from that refuge it should have gotten between the last ice age and now. And so you see plants that move about 100 meters per year might have gotten up to the Nordic areas. Plants that only move 50 meters per year would not have. And in order to get to the top of Norway, plants have to be moving about 200 meters per year. And so there's some idea that some species that we see with a northern range limit, that may not be determined by physical environment. It may not be determined by the fact that it's so cold up there that they can't survive. It may just be determined by the fact that it was glaciated and they couldn't live on top of the ice, but then they haven't had time to sort of move back up to that area since the last ice age, and they're still basically moving up. All right. So all of this brings us to the definition geographic range, or a geographic range. And this is the entire region over which a species is found. There's actually a lot of wide variation in geographic range. So some organisms are found in very, very, very small ranges. A good example of that, at least a local example, is the Venus flytrap. So the Venus flytrap is actually native to a very, very small region in 2 states, one of which is South Carolina. It actually occurs right on the border of South Carolina and North Carolina. And that's the only place where this plant is found in the wild. It's found in a lot of gardens nowadays, but it's only found in the wild in this one little tiny area. Contrast that to the dandelion, and that picture, that map with all the green. Everywhere that's green is where the dandelion is found. There's a county where there are dandelions. And what you'll notice is that there's dandelions almost everywhere, including South Carolina and California and Alaska and Florida. So again, very, very different with these 2 plant species in terms of their geographic range. Now, what you probably noticed from the map of the dandelion geographic range is that populations have very patchy distributions. And this is true at a large spatial scale, for example, over the entire US, where we see some counties with dandelions and some counties without. But it's also true at very small scales. For instance, in your backyard, you might look out and you see dandelions, and they're clumped in some areas, and there's no dandelions in others. So at the large scales, typically these patchy distributions are determined by climate, by the amount of rainfall, by the temperature, how much frost you get, when you get the frost, that sort of stuff. At a smaller scale, it's pretty unlikely that there's differences in rainfall or differences in temperature across your backyard, right? And so at the smaller scale, the things that matter in terms of this, where you find the organism and where it's not present, are things like topography, where the water pools and sort of soaks out the roots, or where it dries out too fast and there isn't enough water, or soil type. You might have a pocket of nitrogen-rich soil in one area, and that allows a plant to grow in that little patch. Biotic factors. Again, I might have sort of root herbivores in one area and not in another, and so that excludes the plants from one location. All right. So when I'm thinking about patchy distributions, and particularly patchy distributions on a more local, small scale, not across a whole map, not across a whole country or continent, I can talk about dispersion, which is the spatial arrangement of individuals within a population. And in general, there's 3 types of ways that individuals can be arranged, 3 classes. The first is regular dispersion, and this is where individuals are relatively evenly spaced throughout the habitat. And you can see an example of that in the center figure. So, there's about the same distance between each organism and the other organisms around it. An alternate spatial arrangement is what's known as random dispersion. And this is what you would get if you sort of closed your eyes and threw darts at the surface. It's just where they land, and that's where they live and grow. And so that example is shown on the right. And then we have clump dispersion, where individuals are actually more grouped together than they would be by chance, by just where you might throw darts if you were blindfolded and throwing darts on a board. So why do you get these different spatial arrangements? Well, typically you get clumped dispersion, either because of distribution of resources. So in that case, the resources themselves might be clumped. I might have a patch of nitrogen-rich soil here, and so the plants grow there, and then it's nitrogen poor right next door, and then there's another clump or another area of nitrogen-rich soil, and the plants grow there. You can also get clump dispersion because of limited dispersal. So for instance, if I have a mom plant, and she makes some seeds, and the seeds don't go very far, then all the little babies are going to grow right in around her, and so you can get these sort of clumped patterns of where the plant's growing just based on the fact the seeds aren't getting that far away. You tend to get random dispersion based on the distribution of resources. So frequently resources are kind of scattered over the landscape just by chance, as if someone randomly threw them out there. And so in that case, you will get a random distribution of organisms that require those resources sort of spread out where those resources are. Typically, you get regular dispersion as a result of competition. So you can imagine a plant or an animal basically defining a territory. Maybe the plant uses up all the nitrogen or uses up all the water within an area around itself, and so nothing can grow close to it. You can't have clump dispersion because there's not enough nutrients. Or maybe a bird uses up the one nesting site in a region, and so that there's no other birds that can be near it because that nesting site is already used. All right. So that's spatial arrangement. We can also think about where organisms exist based on what we call the ecological niche. The ecological niche is the combination of abiotic and biotic conditions that a species needs to grow, survive, and reproduce. And ultimately, this ecological niche determines where in space and where in time the organism is found. When we talk about ecological niches, though, we have to be a little careful. Sometimes things are found in places where you would expect them, and sometimes they're not found in those places. And so this leads to the idea of sort of a more narrowly defined definition of niche, the fundamental niche and the realized niche. So the fundamental niche is everywhere that an organism should be able to grow based on the abiotic conditions. The realized niche is where the organism actually does grow or actually is found, and that's frequently different from the fundamental niche as a result of species interactions. So back to that barnacle example, the fundamental niche for the brown barnacle will be everywhere from the tide line all the way up to the high tide, the low tide line all the way up to the high tide line. It should be able to grow anywhere in there. That's its fundamental niche. That's where the physical environment says it's okay. But then the realized niche is actually just that higher up area, that smaller brown area, where basically it can grow, and it's actually found there because it's not excluded by the blue barnacle. Scientists often use what's known as a niche model to predict a species' range. So to try and predict where on a map that species will be found. And we saw that with that example where the scientists were trying to predict the range of plants in Australia based on abiotic factors. What's important about these niche models, almost all niche models, try to make their predictions based on environmental conditions, based on the abiotic conditions. And as a result, almost all niche models predict where a species should be based on fundamental niche, not based on a realized niche. So often these predictions are wrong because the scientists aren't taking into account species interactions or biotic effects. And that's a well-known problem with niche models. All right. So that's sort of how we define populations and where they're found and sort of good metrics for characterizing different populations. And population distributions as well. Next, what we're going to talk about is abundance and how we estimate abundance. So a number of different techniques that scientists use to figure out, hey, how many organisms are in this location at this time? One common way is what's known as area-based counts. And this is really good for non-modal, immobile, not moving organisms. It can also be used for aerial surveys of mobile organisms. For example, if you're up in a plane and you take a picture of sort of moving animals, you can use that. Basically what you do is you throw down a series of sample plots. So this is what's known as a plot-based method. Frequently we call these plots quadrats. So maybe I'll throw a quadrat down there. And then you count the organisms inside the quadrat. So I might count here and I get 1, 2, 3, 4, 5, 6, 7 urchins in my plot. I might throw down another plot and count again. 1, 2, 3, 4, 5, 6, 7, 8. There's 8 urchins in that plot. And I throw down another plot or another quadrat and I get 1, 2, 3, 4, 5, 6, 7, 8. 8 in that one. So on average, if I average over my 3 plots or my 3 quadrats, I find that there's about 7.67 urchins per quadrat. So then what I can do is I can say, well, if there's about 7.67 urchins per quadrat and my entire area has 12 quadrat-sized squares in it, then I would have 12 times 7.67 or 92 urchins in that whole area. And in fact, if you actually add it up or count it up, there are 95. So that's a pretty good estimate. And I didn't have to count every single urchin. A couple of assumptions go into using area-based counts. First, you have to assume that you counted accurately. That seems like a no-brainer, but sometimes organisms are camouflaged and it's hard to see them, and so you might get your counts wrong. Another big and maybe more subtle assumption is that quadrats have to be good representations of the entire area. So in this particular example where I estimated 92 and there were 95, my quadrats were probably pretty good representations of my full region. But what if my urchins had been distributed like this? Well, all of a sudden those same 3 quadrats would not be very good representations of the entire area because there's large chunks of that area with no urchins in them. And so in that case, that assumption would fail and I would actually get a very bad prediction. I would have predicted 92 urchins and I would have had substantially less in this area. So when I'm designing area-based counts, there's a couple of things I can think about or I can do. First, you might wonder, well, how do you put down the quadrats, right? Where do you put them? You can put them down randomly, wherever you just happen to throw them, or you can put them down evenly spaced. It doesn't really matter. As long as you're not sort of purposely putting them in areas that are higher or lower density than the average area. Then the next question you might ask is, well, how many quadrats do I put down? And obviously, if I had to put down 12 quadrats, I would have covered the whole area and I would have counted every urchin. The more you put down, the more accurate you are. If I counted every urchin, I would have gotten 95 and there were 95. But it's sort of a trade-off between how much time and money you have and how accurate you want to be. So basically, when you're making a design using sort of area-based counts, you want to put down as many as you can afford, but no more. And the more you put down, the more accurate your estimate, but also the more cost and time involved. All right, so that's one method. That's a plot-based method. The next methods we're going to talk about are plotless. There's no sort of plot we're throwing down. There's no quadrat. And these are known as distance methods. And these are good for both mobile or sessile organisms, so things that move or things that don't. And these typically rely on what's known as a detection function. So the simplest approach is what's called a line transect approach. And this is where the researcher or the observer walks along a transect line. So they might walk along this urchin floor. And then they're going to count each individual that they see. And so maybe I can see for 1 meter on either side of me, and I'm going to count all of the individuals in that meter. And what you'll notice is this makes the assumption that I can see perfectly out to a meter and then I can't see anything beyond that, right? And so that is what's known as my detection function. I basically say, well, I detect up to a meter on the left and I detect up to a meter on the right, and I don't detect anything outside of that. And so when I have a sort of square-like pulsed or step function detection, it's pretty easy to sort of do this. I can sort of count up all the urchins in there, and if you do that, you'll find that there are about 30. And then you can say, okay, well, you know, I was able to see for 2 meters on either side over this strip, and it turns out that my entire region has 3 strips of about that size, so I would estimate that there are 90 urchins in this field, and we know that there were 95. So again, that's pretty accurate. It gets a little bit more tricky when you don't have that sort of step function or that pulse detection function. So in reality, I might be able to see out to a meter on one side and out to a meter on the other, but my ability to detect organisms at a meter is probably less than it is at half a meter or at a quarter meter. And so when that's the case, you have to have distance measurements and you have to determine what your probability is of detecting organisms at different distances. So we're not actually going to do any examples like that, but realize that frequently when we're doing these line transects, there is this detection function that is not sort of a pulse. It's a little bit more complicated. And so in that case, you need to actually measure how far away the organism was that you detected as you go along the line. All right. Another method, or another plotless method, is what's known as point sampling. So that was the line transect approach. Another method is point sampling approach. So in this particular case, again, we're going to walk along a line, and either at random points or at evenly spaced points, we're going to stop, and we're going to measure the distance to the nearest individual. So I might be going along this line, and I might say, okay, I'm going to stop at the first red dot, and I'm going to measure the distance to that first urchin that's closest to me. And then I'll stop at the second dot and measure the distance to the closest urchin, and I'll stop at the third dot and measure the distance to the closest urchin. And so maybe the distance to the first urchin was 0.42, to the second 0.34, and to third 0.5. Great. Those are kind of distances. That doesn't tell me abundance, right? You might be wondering, well, how do I get from these distances to abundances of urchin, to how many urchin are this entire area? Well, what you do, the first thing you have to do is you have to convert from the distance measured to the plant to the average area taken up per plant. And so if you say, well, it's about 0.42 meters to the first urchin, then you can assume that 0.42 squared is about the area taken up by an average urchin. If it's about 0.34 to the second urchin I see, then 0.34 meters squared is about the area taken up by an urchin. Same thing, if I measure 0.5 to the third urchin, then I can say, well, on average, urchins take about 0.5 meters squared in terms of the space each one takes up. So that's the average area per plant. Still not an abundance estimate. But if the average area per plant is, for example, 0.42 squared, if I take 1 over that, it's going to give the number, or the average number of plants per area, right? I just kind of flip that. The average area per plant, I take 1 over that, I get the average number of plants per area. And so that might give me 4 plants per unit area, 8.7 plants per unit area, and 5.7 plants per unit area. And so on average, I'm getting about 6.1 plants per unit area. Then what I need to do is figure out how many unit areas I have. So say that quadrat wasn't a meter squared, and I've done all my measures in meters. Then we know we have 12 meters squared in this whole region. I know that on average, each plant takes about there's about 6.1 plants per meter squared. So if I take 6.1 times 12, that gives me an estimate of 74 urchins in that area. And in this particular example, I had about 60. So again, not a terrible estimate. All right. So again, those methods, those distance methods are good for both mobile and sessile organisms. The final estimation method we're going to talk about is mark recapture, and this is best for mobile organisms. So how does mark recapture work? The idea is you basically go out into the field, you capture an organism, you mark it in some way. Maybe you paint it, maybe you clip its wings, you do something so you'll know you caught it before, and then you release it back. And then you return at a later time point, maybe the next day, and you capture organisms again, and you figure out what fraction of the new organisms you've already captured in the past. All right, so let's give an example. So here I have a population of grasshoppers, and maybe I captured one, mark them with a star. This is all on Monday. Maybe it's on Monday. I capture another one, mark them with a star. Capture another one, mark them with a star. Fourth one, fifth one, and a sixth one. All right, so now on Monday, I captured 6 grasshoppers and I marked each one of them with a star. So that was my first day. Then what I'm going to do is I'm going to return on Tuesday and I am going to recapture grasshoppers. So maybe I capture this guy. All right, he was marked. So the first one I got, I'd already captured him before. I capture the second one. Oh, he wasn't marked. So this is the first time I've captured him. Capture a third one. He was marked. Capture a fourth one. He wasn't marked. So on Tuesday, I captured 4 grasshoppers. And of the 4 that I captured on the second day, 2 of them were recaptures. 2 of them I had already caught. Again, you're probably wondering, well, how do I turn this capturing into an abundance estimate? Basically, we use the fact that the proportion that are recaptured on the second day should equal the proportion of the total population that was caught on the first day. So basically, if I take R, if I recaptured 2 on the second day out of a total of 4 that I captured on the second day, then that should equal the total number that were marked on the first day divided by the total population size, which is what I'm after. I'm trying to estimate the absolute abundance of grasshoppers in this field. All right, so if I do that, I put 2 over 4 equals 6 over n, and I solve for n, I get 12, which is exactly how many grasshoppers I had. All right, so I'm actually going to run back here and tell you about the assumptions. So the assumptions when using a mark recapture technique, you have to assume that there's no births, deaths, immigration, or emigration in the time between when you did your first marking and when you did your recapture. If a whole pile of new baby grasshoppers were born, then this technique wouldn't work very well. Or if all of your grasshoppers died, then obviously you're going to go back in and recapture it's going to be a very different population. You have to assume that all individuals are equally likely to be caught. So some animals in particular become trap happy, especially if your traps have food. And so if you keep capturing the same individual over and over because the individual has learned to associate your trap with food, then that can cause a problem. You also have to assume that marking them does not harm them or alter their behavior. So if every grasshopper I marked got killed, this technique wouldn't work very well. You also have to assume that marks are not lost over the time period between when you mark them and when you come back in and recapture them. If marks are lost, then you're going to, again, get a bad estimate. All right. So that's how we measure population abundance. But I want to stress that we could go in and measure the population abundance today, and that could change a year from now, 10 years from now, 100 years from now. So populations are dynamic. They're always changing in size, they're always changing in where they're found, and that's what we're going to end up talking about a lot for the next part of this unit. So populations can change in abundance over time. We saw that with the bald eagles. I had a very low chance of seeing a bald eagle in Toronto when I lived in Toronto. But now when I go back, I've actually seen them. I've actually seen them around Niagara Falls, and I've actually seen them in around the Lake Ontario, right in the GTA, in the Greater Toronto area. And that's because the population of bald eagles has skyrocketed since I left Toronto. Populations change in abundance over space. So I didn't have a high likelihood of finding bald eagles in Toronto because there weren't a lot. But I could have gone to other places in Canada where I could have probably seen bald eagles pretty easily, both on the East Coast, but especially on the West Coast out in British Columbia, which is right above Washington. There is a high population of eagles there. And so if I really wanted to see bald eagles, I should have gone to British Columbia. Also, populations can change in membership over time. So I might have one population of bald eagles on the west side of the mountain and another population on the east side of the mountain, and sometimes individuals will migrate from one location to another and they'll join a different population. So again, populations are dynamic, and that's going to be the focus of a lot of what we talk about in the next couple of lectures. So I hope you enjoyed our first discussion of populations and population ecology, and if you have any questions, I'd encourage you to email me at sbewick@ecology.ca.